In today's video, we are going to be finishing or starting to finish Chapter 8, Neuroendocrinology. This part of the chapter is about sex. We are going to be splitting this lecture into two parts. The first part, we are going to be discussing animal reproductive behavior. We are going to specifically focus in on rats. And then the next part of the lecture that we will do will focus on human reproductive behavior. This is our agenda for today. We are going to discuss different stages of sexual behavior in animals, sexual reproduction, copulation, hormones that are involved in copulation, and some neural circuitry that we find in males and females, particularly in rats, that support sexual behaviors and reproductive behaviors. So first, let's start with sexual behavior. So there are 4 stages of reproductive behavior in animals. The first one is sexual attraction. Then we have appetitive behaviors, copulation, and post-copulatory behaviors. So we're going to focus in on each one of these one by one. The first stage is sexual attraction. So during sexual attraction, this is all about bringing the male and the female together. It is tied to with physiological readiness. It can also be shaped by learning. There are different cultural influences of what is sexy and what is not sexy, sexual experiences when people are young. And as it relates to animals, there are different signals that can help bring males and females together, and we're going to talk about what some of those are. So how do we study sexual attraction? How is this something that can be measured in a lab? How do we do this experimentally? One way that we can do this, particularly for animals, is that we can measure the speed of approach, or how fast these animals approach one another in a lab setting in order to engage in reproductive behaviors. We can also measure how hard an animal is going to work to gain access to a potential mate. So maybe what are like some barriers or obstacles that they're willing to cross, activities that they're willing to do in order to access a mate. We can also measure which features are more attractive, and we can do this by manipulating appearances. So we can maybe remove or add certain markings on an animal. We can change in humans. Maybe it's looking at different eye colors, looking at different colors of the hair, different body sizes. We can look at that in both animals and in humans. There's a few different ways that we can study sexual attraction in the lab. So this was a little bullet. I already talked about this. We can manipulate the appearance of someone or something in a few different ways. So what are some examples of sexual attraction? So the first example I want to talk about is male primates. So male primates are attracted by the sight of sex skin. For example, this is a baboon right here. So baboons are attracted to females that have really brightly colored rumps. This area on the females you can see right here. It is going to swell and turn really bright when a female's ovaries are secreting estrogen. So that is kind of a outward signal to males that this female is ready to reproduce, is ready to mate. and it is something that is attractive to male baboons. Second example, also with male primates, they are attracted by particular feminine odors. Um, these odors can reflect how much estrogen that a female is producing, and higher levels of estrogen are associated with a female being able to A, even get pregnant, and B, have a successful pregnancy and having successful offspring. So both of these examples are signaling estrogen. So that is very important. Estrogen plays a very big role, estrogen and testosterone, in reproductive behaviors and signaling physiological readiness. Estrogen secretion in females is related to the release of eggs, and the detection of estrogen levels leads to more successful timing. So, what do I mean by timing? Of course, there are windows of time that women during their ovulation, for example, you're secreting estrogen, eggs are coming down the fallopian tube. There are certain times in a woman's cycle or a female cycle, even if we're talking about a female animal, where they're going to be more likely to be able to get pregnant and carry an offspring than others. So, if males are able to detect these levels of estrogen either via odors or by seeing something, some outward appearance, if they're able to tap into when that estrogen is high, that is going to help them be able to have more beneficial windows of time where they can mate with that female and they can have a successful offspring. Higher levels of estrogen are associated with females changing color, so their body can change, the skin can become swollen. The female body can give off certain odors when they are higher in estrogen. When a female is higher in estrogen, they are more likely to be fertile. And if we have high estrogen and there is copulation, there is a better likelihood of being able to fertilize the egg and then have a successful offspring. So males want to know when females are high estrogen for a lot of different reasons. In most species, female signal either via outward like swelling or changing colors of skin or from odors. Female signaling is not enough in order to have a mate, in order to have copulation happen. There is an aspect of female choice. So females can find one male attractive and mate with them. They can also find another male unattractive and refuse to mate with them. So yes, there is this female signal, and males will approach females, especially if we're talking about animals. But that doesn't necessarily mean that those animals are going to for sure mate just because that male has picked up on that signal. There is an aspect of female choice. You need active female participation in order for copulation to take place. So that was stage 1. Now we are going to go into stage 2, which are appetitive behaviors. If both male and female are mutually attracted to each other, then they are going to show what we call appetitive behaviors. Appetitive behaviors are behaviors that establish and maintain and or promote sex happening. When female mammals are proceptive, that means that they are going to approach males and they are going to remain close to those males. Or they can do this behavioral pattern, which we have seen by studying mammals. Some mammals will approach, retreat, and then approach again. But it's kind of specific to whatever species that you're talking about. This is also a behavioral pattern that we have seen from proceptive females. We can get specific and look at proceptive female rats. So if a female rat wants to display that she is proceptive, she will engage in ear wiggling. So these cute little ears will start to move back and forth. And they will engage in this behavior called hopping and darting. So they will kind of hop close to the male and then they'll retreat. They'll like dart away and then they'll hop back close. So those are two different behaviors that females can show to the males that they are proceptive. So there's a difference in male appetitive behaviors. Male mammals stay close to females. They will often sniff the female's face and or her vagina. An example we have are male birds. Their appetitive behaviors can look like them building nests. They sing songs. They can dance. So here is an example right here. I thought this picture was funny. So these are two different birds. This is the male and this is the female. So this male bird is putting on this dance. He's ruffling his feathers. So he is showing appetitive behaviors. And he is showing that he is ready to mate and is hoping that this female will decide to engage. If you look up birds of paradise, those are not just a plant, they're an actual bird. Those birds, the males are very, very brightly colored, and they do this really interesting dance, and they just caught it on film not too long ago, a few years ago, and it is in those Planet Earth videos, but those are pretty funny. You can find those on YouTube. So like I said, male birds, they'll build nests, they'll sing, and or they dance, just like this bird is doing here. So we have that initial attraction happen, and then males and females are going to show appetitive behaviors. So if all of those things happen, then we will have copulation. So again, if both male and female show appetitive behaviors towards each other, that is going to result in there being copulation. So when we have copulation occur, it is characterized by intromission. So this is when the male inserts his penis into the female's vagina. This is followed by copulatory stimulations, which is usually pelvic thrusts. These stimulations are going to reach a threshold, and then we are going to have the male ejaculating. After ejaculation, we have a refractory phase. So after copulation, we have no mating. This can last from minutes to months. We have seen that refractory phases can be shortened. This is something known as the Coolidge effect. So we see this in mammals. We've also seen this in humans. as male humans also have a refractory phase. So this is when there is a new partner introduced. There will be a shorter refractory period. So that is what the Coolidge effect is. So copulation occurs usually by female choice. A female chooses whether or not copulation will take place. The female is said to be receptive, or the female, if we're talking about animals, she is in heat or in estrus. For most species, females are only receptive when they are fertile. That's an important distinction to make about the differences between animals and humans. So most species will only mate when they are fertile. This is when they are more likely to produce offspring. It is usually when those females are ovulating. Some animals are seasonal breeders, so then females will only be receptive during their breeding season. Some are only one-time only breeders. Salmon and octopus, for instance, only reproduce one time and then they die. So they do not reproduce multiple times. So there are differences in the windows of time in which species breed. Stage 4 are post-copulatory behaviors. They are incredibly variable in different species. Some examples, dogs after copulation have a copulatory lock. So a male dog's penis will swell so much that it cannot be removed from the female. This is kind of a way that dogs prevent other dogs from mating with their female, and they're trying to increase the likelihood that their offspring will survive. Hens, one of their post-copulatory behaviors, this also has to do with the female choice. Hens actually have the ability, and we learned about this not too long ago in research studies, have the ability to eject sperm from roosters. So they looked at studies where hens mated with roosters that were kind of lower on the rank. They weren't like as big, maybe they were a little bit smaller. They didn't have as high of like status in their group of chickens. And so if they have a lower status rooster who mounts them and who mates with them, rather than trying to move away from that rooster, because that can actually get very violent between chickens and roosters, they allow for the mating to take place, but then they are able to eject the sperm of any unwanted mates and then continue to mate with other ones that they see fit. So those are two instances of post-copulatory behaviors, and as you can see, they're pretty different depending on what species we're talking about. So now that copulation has taken place, we can move into fertilization. This is when the male and female gametes make contact and they fuse together. So of course, we're talking about sperm from the male and eggs from the female. If the sperm fuses with an egg, it fertilizes and it becomes an ovum, which we call a zygote. So the sperm fuses with the egg, fertilization occurs. We have an organism that has now been created called a zygote. That zygote is going to continue to develop and form an individual. Fertilization can either be internal or external, so it can take place within a body or within an animal, or it can be outside. So we'll look at some differences in those. The first one, external fertilization. This requires an aqueous environment, or it requires there to be water. Most invertebrates and fish take place with external fertilization. Wow, I just butchered that. Sorry. Most invertebrates and fish engage in external fertilization. So what happens is they release gametes into the water, and then those gametes are able to mix. So we have here with the salmon, so the female is going to deposit her ova onto the riverbed, and then a male comes by, he can detect where those ova are, and then he deposits his sperm, and they swim and together. Here's a little close-up. And ideally, we will have these sperm fertilize these eggs, and then these will become little baby salmon, and they will eventually grow, and the cycle will start all over again. That was external fertilization. Now we have internal fertilization. This is for all mammals, all birds, and all reptiles. They all engage in internal fertilization. That's how their bodies are set up. So internal fertilization occurs inside the female's body. There are a couple of different kinds of fertilization that can occur. There's two different forms. So there can be oviparous fertilization, which means that there are hard-shelled eggs being laid. This is for insects, birds, and reptiles. Or we can have viviparous. This is when live young are born. So again, oviparous is when hard-shelled eggs are laid after internal fertilization, and viviparous is when we have a actual live young being born. So a horse would be viviparous, whereas like a chicken is oviparous. Virtually all of mammals are going to be viviparous. So now we are going to focus in on rats more specifically. We have a cute little picture of some rats on here. So how do rats do it? Most rodents do not show courtship behaviors at all. They don't tend to stay together after copulation, and they are attracted to each other through different odors that they give off or different smells. Female rats ovulate every 4 to 5 days. During ovulation, during this window of time, they are going to seek males, and they're going to display proceptive behaviors. They also, female rats will not stay together after copulation. They are, like we said, attracted through odors. So now we have male rats. And male rats kind of go off of female proceptive behaviors. So the proceptive behaviors in females can cause the male rats to mount the female from the rear and grasp her flanks with his paws and rhythmically thrust hips Um, this is before intermission, so they're going to start thrusting right before intermission. They're mounting the female. Both male and female rats are going to be vocalizing to one another, but these are frequencies that are below our detection, so we don't hear it, but both of the rats can. If a female rat is receptive, it is going to engage in this posture called lordosis. So this is what lordosis looks like right here. This posture or stance allows for intromission. So the female rat is going to arch her back. Her head is going to be pointed up, and she has kind of her rump turned up so that she can be open for intromission. Intromission, of course, is when the male inserts the penis. He does a single thrust and then he jumps off. This is again how rats do it. So he's going to insert, thrust, jump off, and there are going to be multiple instances of this before the male is able to ejaculate. Multiple intromissions is very necessary for rats. They have to do it in this way, and a lot of it is because the stimulation of the female's genitals cause the brain to release hormones. And then these hormones that are being released in the female are going to help support pregnancy. So we have to have these multiple intromissions in order to stimulate the female so that she can release these hormones to support pregnancy and to have successful offspring. Rats do have a refractory period. So the male and female will groom each other. Then they kind of go their separate ways and they ignore each other for about 5 minutes. Then after this window of time, the female is going to start showing proceptive behaviors again. And then this whole pattern is going to repeat. There are certain hormones that are implicated in copulation. So the first one is testosterone. So we always abbreviate testosterone as TTT. And this mediates the rat's interest in copulation. So testosterone is very important for males even being interested in wanting to reproduce and wanting to copulate. If we have a castrated male, so castrated means that we have removed the testes, we will see that that male ceases to ejaculate and ceases mounting behaviors. So we will see a total extinction of those behaviors. However, if we treat these castrated rats with testosterone, we inject them with testosterone, we will see those behaviors resurface again. However, it is not dependent on the dose of testosterone. So what do we mean by that? It does not matter if we were to give this rat a super high dose of testosterone or even a little bit. Giving them a low or a high dose, we're not going to see any more or any less of a behavior. It's kind of like all or none property with action potentials, right? You either have an action potential or you don't. It doesn't matter, how large the stimulus is. Same thing with testosterone. It doesn't matter how much there is in the body, even just a little bit will restore those behaviors. Adding more testosterone, you're not going to see more of those behaviors. So it's not dependent on the dose. The amount does not predict the amount of mating. So males are impacted by testosterone, and females are impacted by estrogen. So as we have discussed, estrogen is really high during ovulation. So we have high levels of estrogen and also high levels of progesterone. So our hormones, or females' hormones in rats, are surging during ovulation. And this hormone surge increases proceptive behaviors, and it activates the receptivity of the females towards the males. So it is going to make sure that having levels of estrogen in females helps ensure that those females will engage in lordosis and other kinds of sexual behaviors, just like with males with testosterone. And they're going to be more receptive to engaging in copulation. So we're getting at how the brain really regulates reproductive behavior. There are certain hormones that help animals, especially rats and virtually any other animal and humans, to engage and regulate reproductive behavior. There are, in rats, certain neural circuitries that we have found in females and in males that are very important for rats copulating and engaging in certain behaviors. So steroids act on nuclei in the hypothalamus. In females, we are talking about the ventromedial hypothalamus. So this is a cluster of nuclei that are in the hypothalamus. In this area, the VMH, we have found that there are receptors for estrogen and progesterone. What happens if we lesion this area? What happens if we lesion the VMH, if we cut the connections or damage it in some way? We see in females, if we lesion the ventromedial hypothalamus, we are not going to see those lordosis behaviors. We're not going to see the females show receptiveness. They're not going to arch their back. They're not going to signal that they're ready for intromission. So this is really important with females, that the VMH, lesioning the VMH is going to abolish behavior, particularly lordosis. So that is how it works in female rats. What about males? So again, there are steroids in the nuclei, steroids that act on the nuclei in the hypothalamus, very specific nuclei. In males, this is the medial preoptic area, or MPOA. Our research has showed us that this area has a lot of receptors for testosterone. And if we lesion this area in males, so if we lesion the MPOA, it abolishes behaviors, but it does not abolish the motivation to engage in reproductive behaviors. Males also have a vomeronasal organ. This is a group of specialized sensory cells that are near the olfactory, and they detect pheromones from receptive females. So not only is the male picking up information from the MPOA that is stimulating the behaviors, but there's also this vomeronasal organ that is assisting the male with detecting pheromones of the females. The axons from this area send axons to the accessory olfactory bulbs, which then send axons to the medial amygdala. And then this sends axons to the MPOA. And this results in experiencing arousal. So these pheromones travel in through these little like sensory neurons that connect with the olfactory bulb, and we've learned that the olfactory bulb is very important in general across animals and in the human brain for detecting smell. And the vomeronasal organ sends these axons from the olfactory bulbs to the medial amygdala, where it finally reaches the MPOA. And that's how these pheromones kind of get detected and integrated in the male rat's brain. And that is kind of what stimulates arousal. And that is the end of our lecture. So that was talking about sexual behaviors, reproductive behaviors in animals, and kind of specifically talking about rats. On our next lecture, we are going to be talking about human reproductive behaviors, and then that is the end of this section. As usual, please let me know if you have any questions on this lecture.